Okay, welcome, class. In response to this COVID-19 pandemic that's sweeping the globe and causing global panic, I've actually had to create 7 videos in the last 2 days. For those of you who have never done an online video like this, that's actually quite a bit of work. So, without further ado, what you should do at the beginning of this video is you should either print out or have the lecture handout on hand so you can follow through the video with the lecture handout and identify key points that are going to come up on the lecture exam. I would also take a look at the learning objectives and go to the study guides and look over the lab and lecture study guides to get a better idea of what types of questions could be asked. Now, your lectures that you get from me from now on are going to be in this format. There's no associated lecture quiz with this particular lecture because I want you focusing for the lecture exam coming up next week. So, without further ado, what are we going to talk about in today's lecture? Well, this is lecture 2 and 3 on Blackboard, and it covers the rest of um, the rest of the concepts associated with the skeletal system that I want you to know from me going into this lecture exam, so everybody in both of my classes is on fair footing. So the first point we're going to talk about is bone formation, and we're going to distinguish between intramembranous and endochondral ossification. Intramembranous and endochondral ossification come up big time when you guys take things like the T's and the HESI. It's one of those key points on those exams, so be expecting questions about bone formation on your exams that you have to take in order to get accepted into a nursing program. Then we're going to talk about bone growth. We're going to talk about the growth plate or the epiphyseal plate, and we're going to talk about what the process of bone remodeling is and what influences that. And then we're going to cap off this lecture with the hormonal regulation of bone growth, and then we're going to focus on mineral homeostasis, specifically calcium homeostasis, bone breaks, and bone healing. So without further ado, let's go ahead and get into it. Bone formation. In utero, bone formation is complete by the end of embryonic development. So from conception to week 8, you have an embryo. From week 8 to week 37 to 42, you have a fetus, which ultimately is born and we think of as being a newborn or a baby. So when we talk about these processes of intramembranous and endochondral ossification, we're talking about the formation of bone that takes place very early during development, during embryonic development. Now, all bone that's formed from membranous connective tissue or mesenchymal connective tissue forms via a process called intramembranous ossification. All bone that's formed from replacing hyaline cartilage connective tissue with bone connective tissue is called endo, which means within. So endo and then chondral ossification. So we're going to look at intramembranous ossification, then we're going to look at endochondral ossification. And remember, these concepts come up again when you take things like the HESI and the T's, which are those tests or exams you have to take to get into these allied health programs. Now, intramembranous ossification, important point number 1. Intramembranous ossification occurs during embryonic development and it forms flat bones. So the cranial bones, sternum, the clavicle, all of those bones are formed during this process of intramembranous ossification. It's a 4-step process, and we're going to go over each step one by one so we get a better idea. As we walk through this video, if you look at the handout, you'll see blank labeling diagrams that you can follow along with the lecture to label. So the first step in intramembranous ossification, which forms flat bones, is you get the formation of what's called an ossification center. So notice that this says mesenchymal cell. Now, if you remember back to the histology section, the connective tissue from which all other connective tissue derives embryologically, meaning in the first 8 weeks of development, is called mesenchymal connective tissue. These mesenchymal cells will differentiate to form a type of cell called an osteoblast, meaning that there's a chemical cue that essentially tells them, hey, we need to start forming bone and you guys need to become osteoblasts. So these mesenchymal cells differentiate into osteoblasts, and these osteoblasts start tacking on immature bone called osteoid because that's what osteoblasts do. They make bone. So whenever you see stem cell differentiating into an osteoblast and these osteoblasts clustering in a specific area, that area in which these osteoblasts cluster is called an ossification center. Now, step one in intramembranous ossification, which forms flat bones, notably the bones of the cranium, is you get the formation of an ossification center when mesenchymal stem cells differentiate to form osteoblasts that cluster and start building bone. That immature bone that they build is called osteoid. Now, those osteoblasts will continue tacking on bone, right, further and further out. So you'll get this bone matrix, and some of those cells will remain in the mature bone. Once a cell is completely surrounded by mature bone, it differentiates to form a metabolically and anatomically unique cell, or a mature bone cell, called an osteocyte. So out here we have osteoblasts kind of replacing that mesenchymal connective tissue with bone. And then, right, some of those osteoblasts remain where they're at. They become completely surrounded by bone. They change their shape, they change metabolically, they change their function, and rather than building new bone like osteoblasts, osteocytes maintain the pre-existing bone in the area. So the osteoblasts in step two continue to secrete that osteoid, that calcified matrix, right? That kind of pre-bone, but you can think of it as just tacking on new bone. And then any of those trapped osteoblasts ultimately convert or turn into osteocytes when they're no longer tacking on new bone but maintaining pre-existing bone. So those osteoblasts start to build out, and you can actually see this if you look at it under a microscope. So this is an in vivo image of bone being formed. And what you're looking at here is a spicule of bone, so just a little shard of bone. And these osteoblasts are tacking on bone, and they're moving from this calcified area out, and you can see that they're replacing this mesenchymal connective tissue. So what you're looking at in this image is intramembranous ossification, and specifically step two of intramembranous ossification, where you have some calcified matrix, right, meaning mature bone, and then these osteoblasts are just building the bone out. Now, you know also this is intramembranous ossification because those osteoblasts are not replacing cartilage with bone, they are replacing mesenchymal connective tissue, a sheet of mesenchymal connective tissue with bone. So when mesenchymal cells differentiate to form osteoblasts that then replace that mesenchymal connective tissue with bone, you're talking about intramembranous ossification. So that bone will continue to build out until we get what's called woven bone. Now, that mesenchymal connective tissue will condense to form the periosteum, and remember, within the periosteum we have special cells called osteoblasts. So those osteoblasts remain there, and they're really important for bone healing and bone remodeling. And then in the middle you get all these vascular networks that the bone grows around to form what's called a woven bone. In the final step, that woven bone is replaced with lamellar or mature bone. We have compact bone connective tissue toward the outside. So we have compact bone connective tissue toward the outside, and then we have spongy bone connective tissue within the inside. The spongy bone connective tissue in a flat bone is called diplo, and in that diplo you're going to find red bone marrow, which is responsible for producing red blood cells, white blood cells, and platelets. These plates of compact bone connective tissue that kind of sandwich the spongy bone connective tissue form the outside, or the tough periphery, of those flat bones of the cranium, and it's typically all we see when we see a bone from the outside. And remember that all bone in that final stage, when woven bone is replaced with lamellar bone, which just means mature bone, that we still have this periosteum, and the periosteum is important, right, in both long bones and flat bones, because the periosteum has what are called osteoblasts, and those osteoblasts can make new bone. They play an important role in bone healing and bone remodeling. So that's intramembranous ossification, and intramembranous ossification is responsible for forming flat bones. Endochondral ossification, on the other hand, is responsible for forming long bones. So when hyaline cartilage connective tissue in the embryonic skeleton, because that's what initially forms the embryonic skeleton very early in development, when hyaline cartilage connective tissue is replaced with bone connective tissue, we refer to that as endo, meaning within, chondral, so within the cartilage, ossification. That's essentially replacing cartilage with bone, and that's how long bones like the femur, the humerus, the radius, the ulna are formed. So when you look at step one, what you get is what's called the primary ossification center. That's essentially where chondroblasts, cartilage cells, differentiate and they actually end up forming osteoblasts. So you get these clusters of osteoblasts that form these ossification centers, right? And then you also get what's called a bone collar forming, but pay closer attention to what I have labeled on your handout. So we have this ossification center and this bone collar that forms. And as bone starts to replace cartilage, you start to get the beginning of the medullary cavity as that cartilage kind of deteriorates in that matrix. After the cartilage at the center of the diaphysis deteriorates and develops that medullary cavity, you get the paraosteal bud invading, and that's when that spongy bone begins to form within that cavity, and that spongy bone is what forms the endosteum within the medullary cavity. So the medullary cavity has yellow bone marrow, and within the endosteum we get a little layer of spongy bone. Now, the most important for me that you guys know is once the diaphysis is largely replaced with compact bone connective tissue, what we retain at the end of these long bones are what are called secondary ossification centers. And these secondary ossification centers will replace the cartilage in the epiphysis of these long bones with bone connective tissue, specifically spongy bone connective tissue. And remember, within spongy bone, there's red bone marrow, which makes red blood cells, white blood cells, and platelets. One of the things that the cartilaginous structures that remains afterward, one is the articular cartilage, which is hyaline cartilage connective tissue, which is nice and smooth and allows bones to move. The other thing that remains after endochondral ossification during embryonic development, through fetal development, early childhood, up until you're done growing, is what's called the epiphyseal plate. The epiphyseal plate is a layer of cartilage that has metabolically active cells that are still growing. And because these cells are still growing and elongating that bone, we refer to the epiphyseal plate as being the growth plate. And with kids, this is one of the easiest places for them to break bone. It's one of the easiest points at which bone can fracture because cartilage is not as durable. It's not as hard as bone is, so it's easier to physically break that. And a lot of kids, if you've ever met somebody who has a leg that's like an inch shorter than their other leg, they may have had a break in their growth plate when they were young that wasn't set properly. So that growth plate remains, and endochondral ossification is a process in which long bones are made. Now you can pause on this video, you can write those things down, you can have them available to you because I'm not going to be traditionally proctoring an exam. I'm going to be asking you critical thinking questions, right? So bone growth and bone remodeling. As you grow, your bones elongate, your bones physically grow, and the growth of bones, especially long bones, that accounts for much of the height we associate as the difference between, for example, an infant, a child, an adolescent, a teen, a young adult, and an adult, and then an older adult. You see that they actually shrink a little bit as they get older. That's because of the intervertebral discs actually getting a little bit flatter. So what we're going to look at first is bone growth. And then we're going to briefly talk about bone remodeling, and I'm going to give you a little bit of a heads up on something that you're going to see on the Unit 2 lecture exam. So if you look at the growth plate, here we have a long bone and we're looking at the epiphysis. This is called the epiphyseal plate. Now, the epiphyseal plate is also called the growth plate. If you see a growth plate in a bone, you can tell me a few things about that organism. If it's a human, you can tell me, well, that person hasn't gotten done growing. If there's still a layer of cartilage in there, that person just hasn't finished growing yet. And it can tell you something about somebody's age, for example, if you're a forensic scientist. So when you look at the growth plate in action, when you look at it under a microscope histologically, it's broken into 4 zones. There's the zone of proliferation, there's the hypertrophic zone, there's the calcification zone in which that cartilage matrix becomes calcified, and that's another way of saying turns into bone, and then the cartilage starts to die as that bone replaces it. And then you have the ossification zone, which is where new bone formation is occurring. Now, what I want you to kind of think about is this as being a race between the cartilage cells undergoing mitosis and the zone of proliferation and the bone cells, right, creating bone in the ossification zone. And essentially, what happens when the epiphyseal plate fuses is during puberty, men get a burst of testosterone, women get a burst of estrogen, and testosterone and estrogen trigger both chondroblasts and osteoblasts, but they trigger osteoblasts to a greater degree. So these osteoblasts start working a little bit quicker, and this zone of ossification starts to tack on bone a little bit faster than you can tack on cartilage. When the ossification zone catches up with the zone of proliferation, what ends up happening is that epiphyseal plate fuses, and once that bone fuses, that long bone is done growing, and what you're left with is what's called the epiphyseal line. So it's essentially a race between the ossification zone and the proliferation zone, and the ossification zone begins to win that race when you get those big bursts of testosterone and estrogen during puberty, even though it triggers both the zone of proliferation and the zone of ossification. The zone of ossification moves just a little bit quicker, and after a few years of that elevated hormone, right, it's going to catch up, and the growth plate is going to fuse, or the epiphyseal plate is going to fuse, and you're going to be left with what's called the epiphyseal line. So there are a number of factors that influence height. There are genetic factors. It's a polygenetic trait, meaning that there are a bunch of different genes. There's nutritional factors. You could have two parents that are 6'7" and you could be 5'8" because you didn't get adequate nutrition as a child. There are hormonal factors. Growth hormone, testosterone, estrogen, thyroid hormone all play a really important role in the growth of bone. Sleep is extraordinarily important because that's when many of our hormones cycle and many of those processes are taking place that allow bone to grow. Exercise, stress, all of these factors influence height. So I am a lot shorter than all of my brothers, probably because one, genetically, I'm not very good. I didn't get a lot of nutritional support. I have a lot of stress. I don't exercise often. And isn't that a weird image? I can't, it's like mesmerizing. So anyway, just look at that for another minute. I'm going to make you look at that for another minute just to bother you. So we've talked about intramembranous ossification, endochondral ossification, and we've talked about growth at the growth plate, right? We've talked about how bone, right, actually grows, how long bone grows at the epiphyseal plate and when they fuse to form the epiphyseal line, and we've talked about some factors that can influence the growth of bone. The next thing that we're going to talk about in accordance with our learning objectives that we're going over today is what's called bone remodeling. Now, bone remodeling is just the continuous process in which old worn out bone is broken down by osteoclasts. The breakdown of old worn out bone is something called bone resorption. So what osteoclasts do, and remember osteoclasts are derived from white blood cells. They're these big multinucleated cells that kind of look like Roomba vacuum cleaners, and what they do is they secrete enzymes that physically degrade the matrix of bone and release that calcium into the blood. So these osteoclasts come through and they break down old worn out bone that you don't want, because bone can get old and stressed out just like anything, right? So the matrix of the bone can start to deteriorate. And once that bone is broken down, these osteoblasts come in and they deposit, or they carry out the process of bone deposition where they tack on new bone. Now, things that can influence bone remodeling in your favor, right? Factors that can influence bone remodeling are nutrition. You got to get plenty of calcium. You got to get plenty of vitamin D in your diet to have healthy bones. You got to get plenty of vitamin C in your diet to have healthy bones for reasons that we've discussed. Vitamin D because that allows you to absorb calcium. Vitamin C because it allows you to, it functions as a coenzyme in the synthesis of collagen, and we need those calcium salts and those collagen fibers to make bone hard and flexible. Hormones can influence bone remodeling. The more testosterone and estrogen you have, osteoblasts are going to be working a little bit harder than osteoclasts, so bone deposition will outpace bone resorption, and bone will be nice and dense, which is something that you want, especially if you're a young lady. You want to tack on that nice dense bone early in life. Exercise. Exercise disproportionately stimulates osteoblasts and it triggers them to tack on new bone or deposit bone, again, promoting things like bone density. Sleep. Growth hormone spikes during sleep, so that can influence osteoblasts and it can trigger them to go to work a little bit harder and to tack on more bone. Testosterone, estrogen spike during sleep, which can also influence osteoblasts. Overall health. Um, is an incredibly important thing, and it's an important thing in maintaining multiple systems, including bone. Bone is one of those things we don't often think about unless we have a problem with it. So, one of the short answer questions to be expecting, and you may want to look into this a little bit because I'm going pretty quickly and I'm just giving you an idea of what to expect on the exam, is describe the process of bone remodeling. And then I may give you some scenario like pretend this happens or imagine this clinical scenario happens. And I'm not going to tell you that ahead of time because I want you to think critically about it on the fly while you're taking the exam. So be familiar with bone remodeling. Now, bone remodeling is just a process in which osteoclasts break down old worn out bone. So you can see in this GIF right here, osteoclasts are breaking down that bone, and then those osteoclasts carrying out bone resorption, physically breaking down bone. So we have those osteoclasts. And then those osteoblasts come in and they replace that old worn out bone. So it's a constant process that's taking place. And make sure that you're able to recognize those cells, describe the process of bone remodeling, and describe some of the factors that influence bone remodeling and how we, as savvy individuals, can influence our bone remodeling in a positive way. Now, we've talked about it, but exercise can definitely trigger mechanical forces on bone, which promote the activity of osteoblasts, and those osteoblasts can tack on new bone, making your bone denser and more resistant to diseases like osteoporosis. Hormones are really important factors necessary for bone growth. Notably among those are growth hormone, thyroid hormone, and your sex hormones like estrogen and testosterone. Androgens are just male hormones, testosterone being the most important. Now, yeah, we'll leave it at that. So, and we've talked about this kind of epiphyseal plate fusion. This just gives you a nice little breakdown of that. And look at that guy. Aren't teenagers just greasy little things, man? I mean, no offense to any of the teenagers in the class, but geez Louise, man. So, one of the final topics in key point 3 that we're going to be talking about is the regulation of blood calcium. Regulation of blood calcium is really important for a number of different things. If I'm going quickly, I apologize because it is 12:30 at night. I promised you I would have this up by midnight, and I have 2 more classes that I still have to convert to an online format. So I'm going to be up pretty much all night. So, the parathyroid glands. You have your thyroid gland. On the posterior aspect of your thyroid gland, you have histologically and functionally unique groupings of tissue that we refer to as being the parathyroid glands. Now remember, the endocrine system and the nervous system are your systems of homeostasis because those systems are capable of detecting, processing, and responding to changes, and they can tell other organ systems what to do. So when calcium levels, this is a stimulus, get low in the body, they're detected by the receptors within the parathyroid glands. The receptors in the parathyroid glands detect the decrease in blood calcium levels. They also process that information, and in response to decreased blood calcium levels, they release their effector. The effector of an endocrine gland is always going to be its hormone. The hormone that's released is called parathyroid hormone. Parathyroid hormone has multiple activities. Secondary activities is that parathyroid hormone works to stimulate reabsorption of calcium in the kidneys and the absorption of calcium along the GI tract, which increases the amount of calcium in the blood. But the primary effect of parathyroid hormone is to stimulate the activity of osteoclasts. So the effector would be parathyroid hormone. The targets would be the kidney, the GI tract, and osteoclasts. The primary target here are osteoclasts. When parathyroid hormone binds to receptors in the cell membrane of the osteoclast, It triggers those osteoclasts to work a little bit faster, which will ultimately release calcium into the blood. As calcium is released into the blood, you get an increase in blood calcium, which in a healthy system will trigger the deactivation of this circuit. And by definition, what we are looking at here with the regulation of blood calcium is a negative feedback system because the response and the stimulus are opposite from one another. If you get a disease state, for example, like hyperparathyroidism, where there's too much parathyroid hormone being secreted at any given point in time, that can actually be really detrimental because what's essentially happening with hyperparathyroidism is that this system isn't turning off when it should, and we're constantly releasing parathyroid hormone. That parathyroid hormone works on this bone. So that parathyroid hormone binds to these osteoclasts and it works on the bone. And what it does is it constantly degrades bone and jacks up blood calcium levels. As bone is constantly being degraded and calcium levels rise in the blood, there's a saying for hyperparathyroidism. It's called stones, moans, groans, and broken bones. So it's weakening the bone. It's making it more predisposed to forming kidney stones because all that calcium is building up. So it can actually be really bad. So be familiar with this general feedback pathway with the regulation of blood calcium because the regulation of blood calcium is really important. The final thing I want you to be familiar with are the different types of fractures. So a comminuted fracture is just a fracture in which the skin doesn't break, and what you have is the bone broken into a series of fragments. Simple fractures, right? This is a simple fracture. A simple fracture is when the bone breaks completely but doesn't pierce or project out of the skin. A compound open fracture is when the bone breaks completely and it projects out of the integument, and you can actually physically see that bone, so that would be a compound open fracture. A compression fracture is just when bone essentially gets smushed. And an impacted fracture is when one bone breaks and then it pushes up into the superior or inferior region of the bone, depending on how that impacted fracture occurs. So briefly look through those. They're not a huge deal on the exam, but I just want you to be familiar with some of the lingo you would hear, for example, working in an ER. And when a bone is broken, because bone is so highly vascularized and it's surrounded by this paraosteum, there are actually very effective mechanisms for healing bone. So the first thing that happens when a bone is broken, let's just say we have a simple fracture here, is that those blood vessels end up forming a big blood clot that, right, doesn't bleed or have this tendency to bleed out if you don't rip the paraosteum. So that paraosteum kind of traps that blood within that tissue or within that bone right there without allowing it to bleed out into the surrounding tissues unless it gets ripped. And what you get is the formation of what's called a fracture hematoma. A hematoma is just a big fat daddy blood clot. Now, fibrocartilage connective tissue then replaces that blood clot, and we get the formation of what's called a fibrocartilaginous callus. That fibrocartilage is then replaced with bone, and we get the bony callus forming, and then that bone is then remodeled, and you'll see that the point at which that bone broke is a little bit thicker and stronger than before it broke because bone is so vascular, it's very effective at healing itself. Now, think about this particular slide when you think about bone remodeling and when you think about factors that can influence the development of osteoporosis, which is a condition of porous bone. And maybe think about these when you're giving me your answer to the short answer question. Think about this graph. Think about these points that I'm bringing up to the right here. Now, that's it. Have a good night. Bye-bye. It's way too late.